Hi guys, my name is Liz. I'm a PhD student at UCL in London, and I'm in Dr. Lauren Burns' lab, who is from Northern Ireland and a big advocate for HD. So I'm going to be talking to you today about clinical trial updates, and hopefully I'll be able to answer as many of your questions as possible. So Huntington was first described in 1872, and it was described originally as chorea because of the prominent motor symptoms. But obviously we now know that it's more than just motor symptoms, it's also psychiatric and cognitive. And we also now know that there's a genetic cause to it. So this gene, the Huntington gene, was first identified in 1993. And this was a really huge collaborative effort involving 58 different scientists and more than 12,000 families, which is like quite unheard of in science for things to be so collaborative. So it's really great and speaks so much about how collaborative the community is. So just to sort of like back to basics on genetics. So a gene is a section of DNA and it acts as like a blueprint for making proteins, which are basically like the building blocks of life. So to make a protein, the DNA is first copied into this message or messenger RNA. And then the messenger RNA is then what's used to like build the protein. And you get two copies of each gene, so one from each parent, and these are called alleles. And so an allele is basically just like a version of the same gene. So in in the Huntington gene, or HTT, there's a section of DNA where the sequence CAG, which is sort of the code in the DNA, is repeated multiple times. And so each of these CAG is called a codon, codes for an amino acid called glutamine in the resulting protein. And so we all have the Huntington gene, and it's the amount of CAG repeats you have within it that determines whether or not this is a mutated gene or a sort of wild type, as you would call it, undiseased gene. So in a healthy or wild type HTT gene, there's 35 or fewer CAG repeats, and for the average person it would be around 17 in the non-diseased population. So this produces the normal Huntington protein, which supports kind of healthy neuronal function. But in the Huntington's mutated gene, there's 36 or more CAG repeats, and this creates a Huntington protein which has a really long stretch of these glutamine amino acids. And it causes the protein to be really sticky, and it basically, within the cell, it will disrupt things and it will lead to neurodegeneration. So now that we kind of know that we have this like target, basically the mutant Huntington protein, and we've known about it for a couple of decades now, we've been able to really expand our therapeutic development to try and not just treat symptoms, but actually slow down or stop the progression of disease. So at the moment it's a really exciting time, and there's loads and loads of drug candidates within different stages of the developmental pipeline. And beyond just targeting mutant Huntington, there's also been a lot of recent advances in targeting what we call somatic expansion. So this CAG repeat within the Huntington protein is also prone to expanding across people's lives. And so the initial length of CAGs that you inherit is really linked to like when disease symptoms will manifest. But beyond that as well, the rate at which the CAG length repeat expands is also really linked to when symptoms manifest. So we, and this is just showing across time, the CAG repeat length expands. And so about 10 years ago, some genetic studies were done to try and kind of understand better how or what determines when symptoms manifest. And so these were called genome-wide association studies. And basically, what they did is they compared the DNA of loads and loads and loads of different people with Huntington's disease and compared it against the age at which these people developed symptoms. And by doing that, they were able to find areas of the DNA and specific genes within the DNA which were linked to either a earlier or later disease onset. And they found that loads of these genes that they identified were involved in DNA damage repair. So when your DNA is damaged, these genes will come or the proteins from these genes will come and kind of repair the DNA. And so we now know that there are some DNA repair proteins which, whilst their role is to repair DNA, they're actually perpetuating this expansion of the CAG repeat. So there's now been an increase in developmental focus in therapies to also target these proteins alongside just Huntington, which traditionally is what we have most of our candidates have been based on. So just also a really quick background on clinical trials as well. I'm sure many of you know, but just in case there's anyone who needs a reminder. So phase 1 trials take place in small cohorts, and they're basically mainly their main goal is to assess the safety and dosage of a potential drug. Phase 2 trials will be in a slightly bigger cohort, and as well as looking at safety, they also aim to assess whether a drug is effective in kind of targeting whatever it is trying to target. So, for example, if it was to lower the Huntington protein, then it will check whether to see if the Huntington protein is lowered. And sometimes accelerated approval can be granted after a phase 2 trial, which would allow a drug to enter the market. But this would almost always also require confirmatory trials afterwards and follow-ups before it can be granted full approval. And then phase 3 trials assess drug efficacy in terms of whether there's actual clinical benefit for the patients taking the drug. Regulatory approval, full regulatory approval, is granted after phase 3. And then phase 4 is in just sort of ongoing monitoring of drugs in much larger cohorts. So in phase 2 and 3 trials, they also often involve placebo groups. So these are groups of people who don't receive the drug but don't know that they're not receiving the drug. And that's just so that we can really confidently compare the drug effect against a matched group of people who aren't taking the drug. But sometimes, instead of using placebo groups, we can use natural history data from observational studies. So as Sorka explained before, these are really, really, really helpful for research. And they're basically just long-term studies of people to see how disease progresses, or how in non-disease populations, how different features progress, so that we can really understand the disease. And so in order for a trial to be a success, you need to measure something. So we measure 3 different things. We measure primary endpoints, secondary endpoints, and exploratory endpoints. And so primary endpoints are the main determinant of whether a trial will be a success or not. So if it meets its primary endpoint, it means that it was a successful trial. Secondary endpoints are not quite so important, but they're really interesting. So there might be things that could support the findings from the primary endpoint, or things that important to explore as well, but aren't going to necessarily determine the success or failure of a trial. And then exploratory endpoints are more for generating new ideas about how to go forwards with new trials, or what we might want to look at next. And they don't really weigh into whether or not a trial has succeeded or failed. So I'll just quickly run through a couple of ones which are going to come up through the trial updates. So I will be talking about, when I talk about target or target engagement, it's basically whether or not the drug is effectively doing what it's meant to do. So in the case of Huntington lowering drugs, like I said, it might be that the goal of it is to reduce the Huntington message or the Huntington protein. So we would measure the amounts of the message or the protein in the blood or in the CSF, the cerebral spinal fluid, which is the fluid that surrounds your brain and spine. And then we might look at clinical endpoints. So the Composite Unified Huntington's Disease Rating Scale is kind of like a composite clinical score that takes into account the motor score, the total functional capacity, and then two cognitive scores, so SDMT and Stroop word reading. And it's a really sensitive measure of basically like symptomatic progression. We might also look at biomarkers. So neurofilament light is a protein which is really important in neurons, and it's involved in like their structure. And we know that when neurons are damaged, neurofilament light, or NFL, is released, and we can measure it again in the blood or in the CSF that surrounds the brain. And this is like a marker of basically your brain's like brain damage or brain health, whether or not your neurons are healthy. And then we might also look at neuroimaging measures. So caudate volume is a really key one. So the caudate is a structure within the brain which is really prone to neurodegeneration early on in the disease course. So it's a really like a neuroimaging marker of focus. And then we also look at safety, of course. And so we'd look at like adverse events or like things that could indicate that a drug is like unsafe. So for the rest of the talk, I'm going to talk through some clinical trial updates with you. So I'm first of all going to focus on Huntington lowering approaches, and then move to a couple of candidates which are moving beyond Huntington lowering. So we'll start with the most recent news, the uniQure results. So AMT-130 is a gene therapy, and it aims to lower the amount of the Huntington protein by lowering the amount of the message. Basically how it works is a harmless virus is injected into the brain, which has the blueprints to make a synthetic message, which is complementary to the Huntington message, and so will bind and prevent the protein from being produced. So AMT-130 is the first gene therapy specifically designed for HD that has made it into human trials. So uniQure began cautiously testing in a small cohort because this like drug approach is kind of a one and done. You have the treatment, it's not reversible. It's meant to last a very long time. Clinical trials are particularly cautious in these cases because it's not like you can just stop taking the pill, you have the surgery and then that's it. So this summer we've had the top line 3-year results from the phase 1/2 trial. So all data here was compared to the Enroll HD study. So the really big one is that the primary endpoint of lowering the Composite Unified Huntington's Disease Rating Scale, it was met. So basically people who received the treatment, their progression or their clinical progression as measured by the cUHDRS score was reduced. And it was reduced by 75%, which is obviously a really big amount. And then you can see here, so if I just maybe get my pointer, hang on, sorry. Yeah, so here is Enroll, so the so matched people which were within the observational study and are matched like demographically, so like age and sex, to the 12 participants in the high dose group on the trial. And you can see that across 3 years, the average change in cUHDRS here is much greater for the untreated people than for those who have been treated. Again, just to highlight though, this is only in 12 people, whereas obviously in the control it's loads of people because this was such a huge observational study. So, and you can see here as well, this is a graph showing, so in grey, again, it's people who were kind of on the matched observational study, who didn't receive treatment, and you can see across the 3 years, each time point, their clinical deterioration as compared to people who received the high dose of the AMT 130. And then supporting this as well, they also looked at other clinical measures. So as I said, CUHDRS is composed of 4 different clinical subdomains, and they found that across each of these subdomains, there were trends for improvement reduction of the rate of progression following treatment. And on top of that as well, they also looked at neurofilament light. So as I said, this is like a marker of neuronal health. And they found that, for someone who has Huntington's disease and is not taking any treatments, you would expect NFL to increase by a rate of around 12% every year. So you expect NFL to continually be elevating with the disease. And they actually found that in both the low dose and high dose groups, that the NFL after 3 years was lower than it was at baseline. So not just the NFL rate of increase is slowed, but it's actually dropped below what they started with, which again is really supportive that there is kind of an efficacious effect of this drug. And they also found that there were no serious safety events since the end of 2022. And so whilst this is really, really exciting news and a really great proof of concept of lowering Huntington's protein being clinically beneficial, this was obviously in a really small select cohort. At the moment, it's being expanded into more people, or cohorts are being expanded, and also the selection criteria is being expanded as well. And on top of this as well, gene therapies are really expensive, and there's a lot of difficulties with upscaling them. So together, all of this means that it's sort of, whilst this is really, really promising, it's still sort of quite a long way off before we're probably going to be able to see the benefit of this therapy kind of like here today. But as I said, this is like huge for all Huntington lowering therapies. So it's proving that lowering Huntington can be clinically beneficial. So as I said, next steps, expansion of the ongoing study cohorts, and there are plans to submit an application to FDA in 2026 with potential for accelerated approval to be granted in 2026 in the US. And uniQure have also expressed that they are open to conversations with the EMA as well, which is the European Medical Association, which is like the drug regulatory board over in Europe. So FDA is USA. And as well as uniQure, there's another gene therapy which is earlier on in the developmental pipeline. So this is RG6662, formerly Spark 10001. And so this is Spark Therapeutics has recently joined Roche, and they've just kicked off a really small safety study which began in June for a similar Huntington lowering gene therapy. So now moving on to another class of Huntington lowering drugs, so antisense oligonucleotides. So going back to the gene to message to protein, so antisense oligonucleotides work by, similarly to AMT 130, binding the message which prevents the protein from being produced. But rather than being like a one and done injection into the brain, these are injected into, or intrathecally, so into your like spinal fluid. So Wave, I'm going to be talking to you today about Wave Life Sciences compound. So they're, so you guys might have all heard of, or is a really well-known ASO is the tominersen ASO from Roche, which was allele non-selective. But I'm going to be talking to you about the Wave ASO, which is allele selective, which means that it targets just the mutant Huntington. So it's only lowering mutant Huntington protein and not lowering as well the like normal wild type Huntington protein. So this is a first-in-class allele selective candidate. And so basically it works by between different alleles, there's like down, just further down from the CAG repeat, there's like a little unit of the DNA which is different between people, and there's a few different like variations it could be, and so basically they are making like a message which is kind of binds to that specific little bit, and means that you can target just the mutant Huntington and not the wild type Huntington. And so they have, this is sort of a press release from last summer, but they completed their Select HD trial, which was designed to demonstrate that not only would their ASO knock down the mutant Huntington, which it did by 46%, compared from the controls to the treated, but also that it didn't knock down the normal Huntington. And then they looked also at NFL again, so looking at like the sort of general healthiness of the neurons, and they found that most people, so the grey bar here is basically the NFL levels that you would expect from non-treated people, and the black lines are the people who were on the treatment group. And you can see that most people had kind of as expected NFL levels, but there were a few spikes. So there needs to be more kind of understanding of what's going on here. It's not like everyone had these spikes, but we need to like know what's going on. So there needs to be more analysis here. And oh, sorry, it's not clicking. Yeah, and they also looked as an exploratory endpoint. So just they didn't have the kind of power to be able to look at clinical measures, but they just looked anyway, just out of curiosity. And they saw that there were positive trends in clinical outcomes. So the motor score decreased with treatment. But again, this isn't statistically significant because they weren't powered to do so, but it's just interesting to see the trends which might guide future trials. And they also, on a similar vein, also looked at the caudate volume, so that region of the brain that's really vulnerable in HD, and they saw that the rate of neurodegeneration within that structure decreased with treatment. So again, it's not a statistically significant finding, and we can't kind of base anything off this, but it's just really interesting to see that there are these positive trends associated with the lowering of the mutant Huntington. And then again, safety all looked great, besides the potential for those NFL spikes, which need to be looked into, but in terms of like adverse events and things in the trial, it was all great. So next steps, they're planning a phase 2/3 trial in the US. So this will involve looking to see more rigorously whether the drug actually has clinical benefit. So this will be a really exciting trial. So now moving on to splicing modulators. So this is another class of another class of like a therapeutic approach which can lower Huntington. Basically, when the message RNA is first copied from DNA, in its like original form, it will have loads of kind of units in it which need to be removed before it can be turned into the protein. And this is done by a process called splicing, which is basically just like chopping and changing the message RNA to make it the correct message. And sometimes these little units which get chopped out will have what's called a stop codon in, and basically this stops the production of the protein when it's being read. So in order to make the protein, you need to snip out this bit which has the stop codon. And so splicing modulators work by preventing the splicing and essentially preventing the stop from being removed, which ultimately leads to the degradation of the message rather than turning it into protein. The first drug I'm going to talk to you about is Votoplam, which was previously PTC 518. The phase 2 Pivot HD trial completed in May, and it reached its primary endpoint, which is great, which was to show that Huntington could be lowered using the drug. And they compared a low dose and a high dose to a placebo group. And found that Huntington was lowered. And they also found that there were no spikes in NFL, which is really great, across 12 months, which is a really promising safety sign. And beyond that, they found that the NFL levels at 12 months were lower than at the start of the trial, which again, like in the AMT 130 trial, is a really promising sign that not only is the drug not having a negative safety effect, but it could actually be potentially like neuroprotective for these neurons and like actually reducing the neurodegeneration that's going on. And so again, as an exploratory endpoint, so the study wasn't designed to look at this, but they just looked at it anyway because they had the data. They looked at the clinical scores, so the Composite Unified Huntington's Disease Rating Scale, and they found that at 24 months, the rate of worsening, or the change from baseline, so people who had people who were treated had less clinical decline than those who didn't, and this was dose dependent, so people in the higher group had even less clinical decline than people in the lower group, which is obviously really promising. And again, safety was well tolerated. So the next steps, Novartis has taken over, or they're collaborating with PTC, but they'll be running the phase 3 trial, which is really exciting because PTC is a relatively small company, whereas Novartis is huge. So hopefully loads of money can be pumped into it. And yeah, that's in the process at the moment. So then I also wanted to talk to you about the Sky 0515 splicing modulator, which is from Skyhawk Therapeutics. So this is an ongoing phase 1 trial. So a reminder, phase 1 is looking predominantly at safety, but they're also now looking at like whether the drug is doing what we're expecting it to do in terms of like lowering Huntington. And they found that and so this is just a daily pill that you take, and they found that at day 84, people who took the pill every day on the high dose had a lowering of 62% of their mutant Huntington protein. And this is really exciting because previously, to achieve lowering to that degree, it was only via sort of like intrathecal injection of like ASOs and stuff. We haven't yet seen a pill that's been able to lower Huntington to this degree. And so that's exciting because obviously it's so much less invasive and so much easier to take a pill every day rather than have to go and kind of regularly have injections. So that's great news. As I say, really early doors. This is still the ongoing phase 1 trial. And something else they looked at here was they found that, so basically the effect of the drug is quite non-specific. So whilst it lowers Huntington. There's also kind of other proteins that it lowers, and one of those is called PMS1. And PMS1 is one of the DNA damage repair proteins, which was identified as influencing the age at which symptoms manifest in HD. So this is like super exploratory, and I don't even have any graph that I can show you. I was at a conference a few days ago, and they showed some data, but it was kind of confidential, so I couldn't share it. But they basically found that their drug, as well as lowering Huntington protein by 62%, it also lowered PMS1 by about sort of 20%, which could be exciting because it might have a kind of dual impact of both lowering the toxic protein and also lowering a protein which drives earlier onset through a different mechanism, through this like CAG repeat expansion. But this is really early, and we don't know whether a lowering of 20% of PMS1 would even have an effect on anything. So this is just something that they're going to follow up with. But it's cool to know that this could be another potential kind of avenue. And again, safety was fine. And the phase 1 part C study is ongoing. And they've recently launched a phase 2 study in Australia and New Zealand. And so, as I said, kind of like on this PMS1, so there's like other proteins like this which influence the age of onset of symptoms. And they do this by either slowing down or increasing the rate at which the CAG repeat expands. So whilst there are none currently in clinical trials, besides potentially the Skyhawk candidate, Locus 23 have a small molecule which affects the way a protein called MSH3 acts to kind of cause this like CAG repeat expansion, and they are going to be launching a phase 1 trial soon. And then Regenta also have a small molecule which in animal studies they found that 50% lowering, again this is targeting PMS1, found that a 50% lowering of PMS1 led to a 70% slowing of the expansion rate. And yeah, this was in animals, so preclinical, but there are kind of plans to move forward into clinical trials soon. So exciting stuff kind of on the cards in the future. So yeah, just to highlight, HD Buzz, I'm sure everyone knows it, but as a PhD student, I am on HD Buzz nonstop for their updates and stuff. So if you found anything that I've said today interesting, then I absolutely encourage you to go look on HD Buzz, because it's so packed with all of the kind of like current trials, updates and stuff. And then kind of as Sorka mentioned as well, hopefully Enroll HD2 is going to be able to kind of impact life over here as well, and get a clinic set up here. And yeah, just a huge thank you to everyone. Thank you for having me come speak today as well. And thank you to sort of everyone who's made all this research possible. And I hope to get to chat to a lot of you today as well after the talk. So if you have any questions at all, please fire away.